Hi everyone, I'm going over chapters 10 through 13 of microbiology. This video will cover chapter 12, which is the adaptive immunity. The adaptive immunity is the second line of defense, the first line of defense being our innate immunity. If the innate immunity fails, the adaptive immunity steps in. So the adaptive immunity, it takes a little bit longer to go into effect. It needs to ramp up its resources, but once it does, it's pretty efficient at taking out the invader. It's called the adaptive immunity because our body actually adapts to new pathogens that enter our body. The first order of business is covering the antigen. What is an antigen? Basically, it's anything in our body that generates an immune response. Usually it's like a foreign invader that has entered, and our cells need to be able to recognize that invader and be like, hey, let's go after it. So the reason, the one way to remember antigen is the gen part of antigen, and then think how it generates a immune response. Okay, so the next order of business is epitope. So remember we went over antigens. Here's a computer image of an antigen. Well, on the antigen are like these little parts of it. They're shown in different colors, and these are epitopes. So the epitopes are on the outer part of the antigen, and our cells can actually go up to this antigen and kind of dock with it, and then it's basically like communicating with it and be like, hey, do you belong here? And if it sees an epitope, then it knows, hey, this is a foreign invader, this does not belong here, and then it could cause the immune response to happen. So yeah, that's basically what an epitope is. You could think epi meaning outer, kind of like epidermis is on our outer skin. So the epitopes are the outer part of the antigen that our cells can go up to and recognize. Okay, so we also have in our body antigen presenting cells. They're also called APCs. And there are two types, the macrophages and the dendritic cells. So as you can see, they both have like these tentacle things, and they kind of sit around near our body's openings because they're basically ready to kill and gobble up. They're there to grab these like bacteria or viruses that invade our body, and then after they grab them, they draw them in and they gobble them up. Once they gobble them up, these things actually go into our lymph. Remember in the last video we talked about the lymphatic systems? They go into the lymph, and they're there because we have a ton of white blood cells in our lymph, and then the macrophages, they basically they hand over the antigens and are like, hey, look what I got. Kind of like when your cat kills a mouse, and then the cat comes up to you with the mouse. That's basically what the macrophages are doing, and they do that so that our body can be like, oh shoot, yeah, there are these invaders, we should do something about it. And that's basically, these things are very important because they trigger our immune response. Next sort of business, I want to talk about effector cells versus memory cells. Remember when I went over the lymphocytes? Yeah, there are the two types, the T cells and the B cells. Well, this chapter goes over that the T cells and B cells have two different types called the effector cells and the memory cells. So for example, the T effector cells are the ones, or the B effector cells also are the ones that actually cause an effect to happen. They actually are the ones that interact with the cells or attack the cells, that sort of thing, and cause effects to help stop that cell, the invaders, I mean. So the memory cells are a little different. They don't actually go around killing or attacking, that sort of thing. The memory cells, basically, they sit in our lymph, and they're basically there to remember when we were last invaded by a certain virus or bacteria, that sort of thing. So for example, say when you were 12, you caught a certain virus, and you caused an immune response. Some of those cells attacked the virus, but some of the other cells, they just remember that virus. They're just there to, like, to remember that virus was in your body. Why would you want to remember that? Well, because it's basically because they kind of sit there in your lymph in case that virus ever comes again. It's like, hey, I remember you, and then it already saves a couple steps instead of you having to like, basically it allows your body to more quickly attack that virus because it already basically remembers how to do that. Okay, so going back to the T cells again, I want to talk about how there are two types of T cells, the CD4 helper T cells and the CD8 cytotoxic T cells. So an easier way to remember CD4 is helper T cells because they call for help. And that's what the helper T cells do. When they see the invader, they recognize the epitope, remember, and then they call for help. You can remember the CD4 for help, helper, help. Pretty simple. I had a little bit of fun with this one, the CD8 cytotoxic T cells. I remember that by this little thing here is, I hate Britney Spears' song Toxic. So you could think cytotoxic, the song Toxic, and then you could think CD8, how much I hate that song. Yeah. So I just wanted to show you this because here is what I was talking about, the CD4 and the CD8. So the CD4 call for help and the CD8 cytotoxic. And what's also important to know is that these CD4 and CD8 cells are part of the cellular branch of our adaptive immunity system. So they're part of the cellular branch. Maybe one way to remember that is you could think the calling for help, and you think of, oh, you have your cell phone to call for help. Another way to think of it is these cells actually go out and interact with other cells in our body. They interact with them directly, whereas the B cells, they more so interact with them through an antibody instead of directly with them. So the B cells are not part of the cellular branch, they're part of the humoral branch. And what humoral means, it's basically means our blood, the blood in our body. So the B cells go around in the blood of our body, and then they have these antibodies that they can shoot out to attack the invaders in our blood system. So speaking of blood, remember how I said those B cells were part of the humoral response? Well, those B cells that shoot out those antibodies are also called plasma cells. So you could just think humoral, blood, plasma, B cell. That's basically how an easy way to remember. Okay, so going over TCRs and BCRs, you know how microbiology, it can be really hard to remember different concepts, but this is actually very simple and easy, and I kind of wish more of the vocabulary was like this. So the TCRs are very simply the T cell receptors, and they're found on the T cells, and the BCRs are the B cell receptors, and they're found on the B cells. You really can't confuse these two concepts. What I do find kind of interesting, though, are the shape of the TCRs versus the BCRs. So remember how I said the B cells make the antibodies? Well, this is what an antibody looks like. It's a Y shape, and these Y-shaped antibodies are what kill the invaders. We're not kill them, but they like trap them and stuff and make it easier for our other cells to kill. And anyway, so the antibodies also are on the outer membrane of our B cell, and they act as a receptor. So that's kind of cool. And see, the T cells, each receptor can only recognize one epitope. But the B cells, it's like they have double the chance of recognizing that epitope because they have two binding sites, which is pretty cool. Okay, more about the T and B cells. When they're made in our body, remember the T cells are mature in the thymus, and the B cells mature in the bone marrow. Important to note that the T cells are not don't originate in the thymus. The T cells originate in the bone marrow, but then they travel to the thymus to mature. So when they're maturing, the T cells and the B cells, when they're maturing, our body basically has to go through kind of a screening process to make sure that our immune cells don't attack our human cells, our self, because that would be bad. Like, that would cause an autoimmune response, and like, for example, an autoimmune disorder would cause our cells to attack our organs and our tissue, and that's bad. So our body makes sure that doesn't happen. And there are these things called MHCs, and MHCs are on our cells. All of our cells have them except our red blood cells. And basically, so when an immune cell goes up to one of our cells, it needs to be able to recognize its MHC and be like, oh, no, you are human, or oh, no, you're human too. And so that way it doesn't attack that cell. So if it does not recognize MHC, then that T cell might attack it. So if the T cell maturing does not recognize that MHC, our body actually kills that T cell so that it can't go out there and accidentally attack our cells. So does it recognize MHC? If it does, and then it goes through another screening process, is it self-reactive? If it's yes, it kills it. If it's no, then it passes self-tolerance, and then it can go out into our body. Same thing with the B cells. Does it create antibodies that attack our self-antigens? If not, then it passes. So that's cool. That our body. It sounds weird that our body destroys our immune cells, because like, oh, don't we need lots of them? Why would our body destroy them? But it's actually really important. If it didn't destroy it, the bad immune cells, then our immune cells would destroy ourselves. So that's pretty cool. Okay, so so remember I was talking about the MHCs. There are two types of MHCs, MHC1s and MHC2s. Here's a little chart to help you remember. But remember I said MHCs are found on all of our cells except the red blood cells. Well, those ones I was talking about are MHC1s. The MHC2s are only found on the antigen presenting cells, those dendritic cells and those macrophages. So just think about the dendritic cells and the macrophages having both MHC1 and MHC2. And the way I remember that is MHC1 is number 1, so it's number 1 in our body, that it's on all the cells. And just note that it's not on the red blood cell. Okay. The MHC1 interacts with the CD8 T cells. The MHC2 interacts with the CD4 T cells. The way I remember it, it's the higher number, the 8, interacts with the lower number, and then the lower number interacts with the higher number. I know that's kind of confusing, but I just have to tell myself it's the opposite, that the 8 is the lower number and the 4 is the higher number. And then basically these antigens are presented either inside the cell or outside the cell. So the way I remember that it's on the outside of the cell is I think about those antigen presenting cells. So they basically are like handing over the antigen. So that's why it's on the outside. That's how I remember it. Okay, so now we're going to go over those antibodies. Another word for antibody is immunoglobulin, hence the IG. And there are 5 different types of immunoglobulin, and they spell out GAMED. An easy way to remember that is think game day. So yeah, game day. And if you take the order GAMED in game day, it's the order of the most abundant to least abundant in our body. So IgG is the most abundant, then IgA, then IgM, then IgE and D. Those are the rare. But most of our antibodies are IgG. Okay. Our professor mentioned that we really should remember IgG and IgM. Okay, so make sure you remember those in particular. IgG, they look like they're just monomers, where the IgM they can either be monomers or they can be like these multiple antibodies that make into like a snowflake. So I remember it that way because the M, and I think of like the M in multiple, and so that's how I remember that. The IgM, see how they're shaped like this? Well, like usually like when these antibodies are going around in your blood, they will interact with different antigens and like connect with them, with the epitopes. And with the IgM, look, they could connect with so many different epitopes, so they could like collide with a whole slew of them. And what ends up happening is when it does that, they kind of like agglutinize, which means like they like clump up with the different epitopes. So like it's really efficient at stopping the invader because they could clump them up, and then that invader can't do anything because it's like stuck in like this clump of antibodies. So that's cool. Yeah. The other thing I wanted to mention was the IgMs respond first. So when the invader comes, the IgMs respond first, and the IgGs respond second. So that's just something to keep in mind. Oh, and then I show it here that the IgMs, see, when the antigen is exposed, the IgMs come first, and then the IgGs come second. The way I remember that is I think of M then G, and I think of the word hemoglobin because these things are in our blood, and M comes before G in hemoglobin. This is only when we have the primary antigen exposure. When we're exposed to the same antigen later, like remember how we have those memory cells, and so it could remember if it was exposed again? Well, if it happens a second time, or a further occasion, then the IgGs and IgM immunoglobulin actually come at the same time. It's not like M and then G, they both happen at the same time. And what's really interesting is that the IgG amount is a lot higher than the IgM amount. So remember, primary, M is first, then G is second, but in secondary, they're both at the same time, and then the G has a much higher amount. You might remember higher amount because remember G being the most abundant, so that might help you remember that. All right, so that's it for the video. I hope that was helpful. I know it's a lot of information, but maybe that will help you remember a lot of this stuff. So cool. All right, thanks for watching, and I will see you in the next video.